Hello and welcome to Ask Your Academic. Today's session will cover the MSc Medical Genetics and Genomics. We are joined by our Programme Director, Gerhard May, and our PGT Administrator, Kirsty McCall. Thanks very much for all your questions that have been sent in before today's session. We'll do our best to get through all of those. If you have any further questions throughout the session, you can pop those in the chat box and we'll keep an eye on that. If after the session you have any further questions, you can send them to the administration mailbox and they'll get picked up there. We are recording the session today. That will be sent out probably once all our sessions are complete, so you'll not receive that immediately, but you will get that link. So we'll not waste any time and we'll get started on our questions if that's okay. Our first question we've got, how do we know what courses we're taking and how and when do we register?
Okay, so in the MSc in Medical Genetics and Genomics, there is very little choice, I have to tell you that straight away. You will study a number of courses which are compulsory for this program. There is one teaching block which starts in the middle of February, where you will have a choice of 4 different courses, but as that is in February, there is nothing to worry about. So once you have registered with the university, then at some point you will automatically enrolled on the courses that you will have to take. There is only one course in the first semester that you'll take. That's a really big, fat course for 60 credits. So that's one course that covers the whole of the semester. You'll be automatically enrolled, nothing to worry about.
Perfect. What options for research assistantship or teaching assistantships are there for postgraduate students?
Okay, so this is one of a number of questions about job prospects, really. That's how I interpret this. Because I'm assuming this question isn't about what options are there during the MSc, because I would not recommend that you take on a great deal of paid work during the MSc. A bit of work is always okay, but not a great deal. After the completion of the MSc, our students go usually to, a large proportion of our students go to 2 different directions. One would be diagnostics in a health service. So students would go on to, for example, in the UK, the National Health Service graduate training programs. There is a number of graduate training programs in the different evolved countries. The other direction that students like to go and aim to go for is research. There is always a handful of students with our graduates who go on to do a PhD in some related topic. That may be in the UK, but we've also got students who went off to do PhDs overseas. There are also always students who start working in some technical capacity technician posts. Now, the question asks about research assistantships and teaching assistantships. This is out of the hands of the program director, really. This is entirely up to a student who is conducting or studying for that master's program. It will be your job to look out early and start applying for posts that interest you. What we try to do is help you with this, and you will be allocated a mentor who will meet with you a couple of times in the first half year or so to discuss your aims, your career plans, and maybe give you some advice with that.
Perfect, thank you. What should I prepare for the coming academic year?
Well, one thing we'll do, I have generated a molecular biology revision Moodle. Now, Moodle is a virtual learning environment. You may be familiar with these things from your undergraduate degree. There are different things, Moodle, Blackboard, but I forget what the other ones are called. So once you are registered with the university, you can also, or should also, register with Moodle at Glasgow University. And about 3 weeks before the official start of the course, you will be given access to a revision Moodle by me. So this comprises mostly basic molecular biology concepts, which is hopefully all stuff that you are familiar with from your previous degrees. That is something, though, that I would encourage you to work through when you've got a bit of time, and look at that, work through that. Within the first week of the course, of the program, we are quite mean there. We'll give you a little test, which does not count towards any grades anywhere. So this test is based on that revision Moodle, and we'll use that to look out for students who might need some additional help. So if you do want to prepare yourself beforehand, work through that revision Moodle.
Thank you. And can we get access to the syllabus book for the course?
Now, I'm assuming with syllabus book, you mean a sort of main textbook for the course. If that's what it is, I have to disappoint you. There is no main text, because this isn't an undergraduate program. So we are trying to keep relatively close to what is happening out in the real world in research and in diagnostics. So we don't use a textbook. Everything is based on published papers and on well, material that lecturers will give you. Now, one thing I can do, though, and I'm going to see if I can put this into the chat now. Here's a link. So here's a link. This is a link that me and 3 of my colleagues have for a booklet that we've written about 3 years ago, maybe 4 years ago. This is a freely available PDF file. It's published by the UK Biochemical Society in their publisher, Portland Press. This is a booklet aimed at final year school pupils or sort of early undergraduate students. It's the basics in molecular biology and medical genetics. That is something which I think really won't hurt if you have a look at that to remind yourself.
Perfect, thank you. And can you tell us a wee bit more about the exams? So when they're typically sat, the pattern of the exams?
Okay, what I can do, I can show you something now. You should be able to see an Excel spreadsheet. I cobbled that together yesterday for this webinar. This is, at the moment, the planned assessments for the program. Now, it's not entirely complete because there is this one optional course in February, March that you'll take, and I can't include that one because different courses have different assessment. So at the top here is the first semester from the beginning of teaching in September till Christmas time. That is the second semester just after January till Easter time, and then the third semester down here. Now, there is actually just 2 major exams in the program. One is, where has it gone? This one here on the 17th of April. And this is the main exam that covers the first semester course. There is a second small exam here, the screening exam, that covers one little course which is taught here in the first weeks of the third semester. So here a big exam, here a small exam. All the other yellow things here on that spreadsheet are coursework. So to a large extent, your progress will be assessed by coursework. This, in fact, here, the very first one is a formative piece of coursework that doesn't count for any overall grades, but you will be given feedback, and that feedback would then hopefully help you to submit this year data analysis 1, which is the first piece of coursework that really counts for something. Just for entertainment, I've also put in green here, there is a planned bowling outing, right? So we don't just abandon you to your work and expect you to sit in your rooms and beaver away. Here we'll have an away day. If we can, then we will take the whole class out to an away day at a local castle. This is just an overview. So it's mostly coursework and 2 exams. Actually, maybe I should say here, the first day after the Christmas period, it says class test here. This doesn't count for the exam either, but this is basically a mock exam corresponding to the big exam in April. So this is a class test that allows you to practice the kind of exam that we are setting you, and it gives you an idea how much have you taken in of the first semester teaching. And I should stop sharing again, shouldn't I?
Perfect, thank you. Does the program support additional hours for research versus classes?
Now, I'm a bit puzzled by this question. I'm not quite sure what that means. If you are meaning research during the taught program, then I think there is some time for this, but not a great deal. The first semester course, it's called genetic disease, and that certainly in the first, say, 6 to 8 weeks is very busy. Then it becomes a little bit lighter. I think it should be possible to do a little bit of work on the side, but it will be, again, as I said, it'll be your responsibility to try and identify. Maybe it's possible to find a research group at the university where you can do some research on the side, but I don't actually know of cases where this has happened.
Thank you. Someone would like to know more about any lab work or field work.
Okay, there's no field work, but we have some lab work. In the first semester in this big course, genetic disease, there are a relatively small number of lab work days timetabled, where in the first sessions you'll be practicing again some basics like generating solutions and making sure their pH is correct, and pipetting, because our experience is that some students haven't actually had much experience in a molecular biology lab. And I suppose that now, after 2 years of pandemic, you guys are most likely in a situation where you've had even less lab experience than our previous students have had. So then in the first semester, you'll do a brief practical where you'll use PCR to try and identify the gender of a person for whom you will be given a DNA sample. So you'll get a DNA sample and you are supposed to identify was it a male or a female. based on a PCR technique. In the second semester, all of you will do a course called SNP assay design. This is a lab-based course. It goes over 8 weeks. That's how it's timetabled at the moment. I think it might actually only be 6 weeks now. So that, well, only I say, this is based only in the lab, where you will design your own genotyping assay, and you will then genotype a number of DNA samples. I think it will be about 30 DNA samples that you'll be given, that you can genotype for a particular single nucleotide polymorphism. And if you don't know what a single nucleotide polymorphism is, then don't worry, we'll tell you that really quite early on in the course.
Thank you. Is there much involvement of bioinformatics on the course?
Relatively little. You'll be given a tutorial on the use of software like BLAST, which looks for sequence similarities, but that's about it. There is involvement of bioinformatics to some extent in one of the optional courses. So if the pattern is the same as in previous years, there will be 4 different optional courses in February that you can choose from. One of them is associated with our program a bit more closely than the other 3. And I know in this one course, there will be the use of some bioinformatics tools to analyze next generation sequencing data. The other 3 optional courses will use similar tools, but based on other questions.
Thank you. And what are the PhD prospects like after completing the MSc course in medical genetics and genomics?
Okay. Since the financial crash in 2008, funding for research has dropped worldwide. I think it's fair to say that it hasn't gone back to where it was before. So there are not as many PhD places out there as there used to be, say, in 2007. Getting a PhD place is competitive. However, there's always a small number of our students who go directly from our masters into a PhD. So this is definitely possible, but you need to be absolutely clear that gaining a place on a PhD program is very competitive. You need good grades for this. So when you come to us, you'll basically have to, and this is what your aim is, doing a PhD and going into academic research, or maybe in industry, then you will have to get good grades from the word go.
Thank you. And that's all the questions on my list. I'm just going to look at the chat box now just to see what we've got in there.
Okay.
So somebody has asked, is there any topics in mathematics that I need to be thorough with before the term commences?
I expect a student who aims to do a master's in medical genetics and genomics, in any biomolecular science topic. I expect students to have a reasonably good level of numeracy. We will actually do some training in numeracy. This is the basic lab numeracy that I'm talking about. So this is the question of if you have a particular molecule and you have where you know the molecular weight, how much of that will you need to make a certain volume of a given concentration, stuff that you are hopefully familiar with. So basic lab numeracy is something that I do expect, but will give you some extra help and training. There is no harder math that is required.
Thank you. And Could you tell us a wee bit more about the dissertation? Like, when do we have to decide the topic and propose the idea?
Okay, the dissertation, this is the research project that you will conduct in the third semester. So this is a 3-semester, 1-year master's program. The first 2 semesters are all taught courses, 120 credits worth. And in the third semester, you do a research project, another 60 credits worth. That project usually starts in middle of May. And then you are expected to submit a report early August. This year, the deadline is the 8th of August. So the way that this is done in our college is that any potential supervisor is asked to submit possible projects before Christmas time. And then after Christmas, these potential projects get collated into booklets for the different master's programs within the college. And sometime in late February, you will be, I think it's late February, not earlier than that, you will be given a list of suitable projects that are available for your program, for students in your program. You will then be asked to give a list of priorities. So this year, I think we asked for your top 6 priorities, and then program directors in different master's programs go through this together to try and allocate to each student a project, hopefully from their top list. It is also possible to self-propose projects. So if you happen to have good links with, say, a research group in your home university, then it is possible for you to propose a project outside of the University of Glasgow. Is that what the question was, or was it also about topics?
Yeah, no, I think you've covered that. Yeah, the topics. Yeah. Does the university have a placement program?
Some programs, undergraduate and postgraduate programs, have as a compulsory component a placement, 1 or more placements. However, our master's program doesn't have a compulsory placement component. So we can't really help you with finding some job afterwards. And as I said earlier, I don't recommend to try and spend a great deal of time working on a placement or for paid work outside of your study time.
Thank you. Someone's asked, will we have wet research on animals?
You might. Now, this is a medical genetics program, and really that is largely interested in human genetics. However, research is usually conducted first on other organisms, because ethically it's not really that possible to do a great deal of experimentation on humans. I'm being flippant here. So during your dissertation, your project in the third semester, you may well work in a research group that works on some animal model. So that might be mouse, it might be fly, Drosophila, it might be worms. However, it is my experience that master students don't usually, well, you won't be able to do actually the work on the organ, on the mouse themselves, because for that you will need a special home office experimental license, and you won't be able to get that. So you will be possibly working in a research group who uses mice as experimental model, but you might be working with some tissue samples that someone else has obtained for you, or you might be working with cell cultures, or maybe just DNA samples that have been extracted for you already. So I don't think there will be a great scope for work on the organisms themselves.
Thank you. Approximately how many students will be in the program?
This year there are 39. We have had as few as 25-ish. That's about 5 years ago. But in the last 5 years or so, the number of students on the MSc Medical Genetics and Genomics was around 35 to 42. And to be frank, I hope it's not going to be more than that, because then it becomes harder for staff, and you guys get less attention from us.
Thank you. And someone's asking, will the lectures be online or offline, or sorry, on campus? Right.
At the moment, we're planning with lectures and virtually all teaching sessions to be on campus. There may be small number of teaching sessions, lectures that will be conducted online, but that is largely because a significant number of our lectures, especially in the first semester, is given by people working in the NHS health service. They work in a hospital which is away from the main campus, and it's for them, it might be more convenient to give their lectures online than actually travel to the campus. But on the whole, we plan everything to be on campus.
Thank you. I think this might need to be our last question, just because we're coming up to 11 o'clock there. But someone's asked, do we have a genetic counselling course too? If we're aiming for a genetic counsellor, how can we prepare for it?
Okay. Our team, we teach also a master's program in genetic and genomic counseling. However, that is not running this coming academic year. It will be taking in students again in 2023. However, this is extremely competitive. We're only taking in 8 students by interview. Getting onto these counseling master's programs in the UK is not an easy job. There are not that many, and they are very competitive. Within our master's programs, in the medical genetics master's programs that we're talking about, there is no dedicated course on counseling, although we try to constantly get you to think about the ethical implications of what we're talking about, and to consider how would you, for example, if you have a particular genetic testing result, how would you talk to a family about this? But that's not a formal aspect of the program. I should also say that some of our students, as I said earlier, move on to go into graduate training programs in the National Health Service. The National Health Service also has a counseling, at least one counseling training program that I know of, and I know that students from our masters have moved on to do counseling training afterwards.
Perfect, thank you. Well, that is us now at the end of our session. You've done a great job here of answering all those questions that came through the chat box. There's still quite a few in there. So what I would say is, if you've not had an answer to your question, if you send an email to the administration mailbox, hopefully someone can get back to you on that. So thanks again, everybody, for your time. We hope you found that useful. Take care of yourself, and we hope to see you all very, very soon. Thank you. Bye-bye.
Bye.
Hope to see you soon in September in person.